Hello everyone. Today we are going to talk about the anatomy of the prokaryotic cells. We are specifically going to concentrate on bacteria. Let us take a look at some of the shapes and different arrangements for our bacterial cells. Some bacterial cells actually look like stars and some of them look like rectangles. Some bacteria look spirally and they're pretty thin, so we call them spirochetes. If it's spirally but it's a bit thicker, we call it Sporulation. Vibrio, those look like curved rods. When a bacteria looks spherical, we call it coccus. Coccus is singular. Cocci would be plural. If there are two attached to each other, we refer to it as diplococci. If it's a chain of spherical bacteria, we refer to it as strepto, so it would be streptococci. If it's a group of four, we refer to it as tetrads. If it's a group of 8 to 32, we call it sarcinae. And if it's basically a cluster of spherical bacteria, cluster, we would refer to it as Staphylo, so Staphylococci. Bacillus, those are bacteria that look like rods. If you actually have two rod bacteria next to one another, attached to one another, we would call them diplo. So diplobacilli. So bacillus is singular. Remove the US and put an I, it would be bacilli, and that would be plural. So if you have, as I said, two of them attached to each other, you would call them diplobacilli. If you have a chain of bacilli bacteria, you would call it streptobacilli. Some of them actually are known as coccobacillus because they kind of look like they're not totally rods, and they kind of look like coccus bacteria, but they're a bit elongated, so we call them coccobacillus. And so, as I said, if you have two, call it diplo. If you have a chain, it's strepto. If we take a look at the structure of a prokaryotic cell, all prokaryotic cells, they have plasma membrane. They have cytoplasm, which is the fluid inside of the cell. They have ribosomes, and the ribosomes of prokaryotes, we refer to it as 70S ribosomes. Humans and eukaryotes have ribosomes, but we don't call it 70S. It's 80S for eukaryotes. All prokaryotes have DNA. Now, of course, the DNA of a prokaryote is going to look different compared to the DNA of a eukaryote. Most prokaryotes have plasmids, and plasmids are basically circular DNA. It's not attached to the original DNA, and those plasmids usually carry antibiotic resistance genes. So it's pretty important, and actually, not all bacteria have it though. And some bacteria can actually exchange their plasmids, and we will talk about it. Some bacteria have Fimbriae, which are found surrounding the cell, like so. We're going to talk about their functions in just a few. Some of them have those projections known as pilus, or plural would be pili. Some of them have flagella, which is going to help them move, but there are other means of movement, and not all of them have flagella. A cell wall is going to be on top of the plasma membrane, and on top of the cell wall there is a layer known as the capsule. But again, not all bacteria have the capsule layer. Having the capsule layer is usually going to protect the prokaryotic cell even more against the immune system. And so we always say that the capsule is going to add to the virulence of the bacteria, and it's going to definitely play a role in making the bacteria more pathogenic, meaning the bacteria can cause disease if it has a capsule. Bacteria can produce and secrete something known as a glycocalyx, which is a sugar coat, and it would surround the bacteria. It would be external to the cell wall of the bacteria. The glycocalyx could be composed of polysaccharides, just basically sugars. It could also be made out of polypeptides, which are proteins, or a glycocalyx could actually be composed of both polysaccharides and polypeptides. Now, if that glycocalyx is firmly attached to the cell wall, we call it a capsule. And if it's unorganized and loosely attached, we refer to it as a slime layer. We can actually detect the presence of a capsule by doing a negative stain. So a negative stain would allow us to be able to spot a capsule. Now, if bacteria do have capsules, which, as I said, they're going to be attached to the cell wall of the bacteria, those capsules, they usually protect pathogenic bacteria from being phagocytized. What is phagocytosis? Phagocytosis is the process of let's say the immune cells engulfing something foreign or engulfing a bacteria cell. So when bacteria cells have capsules, that actually helps protect them from those phagocytic immune cells that we have in our body or within the host, basically. So therefore, having a capsule definitely contributes to the virulence of the bacteria. And a lot of the bacteria that are actually capsulated, like they have a capsule, they can cause diseases versus bacteria that are not carrying a capsule. Because, as I said, the capsule protects the bacteria from being engulfed by the immune system of the host. It also protects it. It gives it extra protection from harsh environmental factors, so it's able to survive longer. So an example, so as I said, capsules can prevent phagocytosis, which is again the immune system of the host usually has what we call phagocytic cells, and those phagocytic cells, they engulf bacteria, they engulf toxins, they can basically engulf anything that kind of looks foreign. So having a capsule can prevent the bacteria from actually being engulfed by the immune cells of the host. Having also that glycocalyx can help form a biofilm, which is basically the bacteria attaching to surfaces. Bacteria can attach to so many different surfaces, whether it's the plants, rocks, the teeth, skin. So bacteria are found everywhere, and they can attach to so many different surfaces. Having a glycocalyx can definitely protect the bacteria from getting dehydrated and from losing nutrients. So it gives it extra. The bacteria Streptococcus pneumonia could either carry a capsule or some of them don't carry a capsule. The ones that are encapsulated, those are the ones that are going to cause pneumonia because the capsule is going to be protecting the bacteria from phagocytosis. The ones that are not carrying a capsule, those are usually going to be phagocytized pretty easily by the host, and so therefore they're not going to cause pneumonia. Some bacteria have flagella. Flagella are appendages that are attached to the external part of the bacteria, and those flagella, they propel and they help the bacteria basically move and swim. Flagella, they're made out of a protein known as flagellin. Not all bacteria have flagella. Some bacteria move by other means that we are going to talk about. Um, the flagella structure, flagella, by the way, is plural, singular is flagellum. The flagellum structure is different in gram-positive and gram-negative bacteria. So this is kind of how it would look like if it's attached to a gram-negative bacteria. And this is how it would look like if it's attached to a gram-positive bacteria. So there are slight changes in its structure, but the idea behind how a flagellum would help a bacteria basically move is kind of the same. The flagellum is going to start twisting and turning, and by doing that, it's going to help the bacteria move. So as the flagella rotates, that rotation is basically going to help the bacteria propel. There are different arrangements for bacteria flagella. You would notice that when it comes to naming the arrangement of the flagella, one common thing about all of the names is the suffix, which is "tricus." Whatever prefix comes in front of it would kind of give you a hint about how the flagella looks like and how it's arranged. So let's say a bacteria actually has no flagella, it has absolutely none. We say atricus. So A in medical terminology means without. So if I say atricus, that means without flagella. So this bacteria is carrying no flagella whatsoever. If I say peritricus, peri means around. So peritricus would mean that the flagella is basically surrounding the whole entire bacteria. Monotrichus, mono meaning one. So monotrichus is a single flagellum, and usually that single flagellum is just coming out from one pole of the bacteria. If I say lophotrichus, lopho would mean that you have multiple flagella coming out from one pole. Amphitrichus, you have flagella that are sticking out actually from both ends, from both poles of the bacteria. So one more time, you can say atrichus, which means without, peri, which is around, mono, which is a single one, lopho, you have multiple ones coming from one end, and amphi, meaning from both ends, you do have flagella sticking out. And as I said, those flagella would rotate, and that rotation is going to help propel the bacteria. You can see right here a bacteria with its flagella sticking out of its surface. Now, when a bacteria moves in one direction for a period of time, we call that a run or a swim. So you can see right here, the bacteria is moving in one direction. Now, let's say this bacteria wants to turn, it wants to change its direction of movement. You notice that the flagella, they kind of fan out like so, and we call that a tumble. So the bacteria would keep whenever it's moving, it would run and then tumble, run, tumble, etc. Now, those flagella are made out of proteins, and we can call those proteins H antigens. There are actually different types of proteins within the flagella, and we can actually note that by giving it a letter and a number. So, for example, you would say H7, H, and you would give it a number. So the H and the number is telling you kind of like which type of protein is is making up that flagellum right there. So for example, you would find when it comes to naming the bacteria, you would notice sometimes they would say E. coli H7. The H7 part is referring to the flagellum. Some bacteria are known as spirochetes, and those spirochetes, they have an axial filament. Another way or another name for axial filament is an endoflagella. Endo because it's inside. So when we looked at the flagella previously, you saw how the flagella was sticking out of the surface of the bacteria. This flagella right here, or this axial filament, is not sticking out of the surface of the bacteria. It's actually embedded inside of it. And so that's kind of the biggest difference between a flagella and an endoflagella. And that endoflagella, or that axial filament, it's made up of bundles of fibrils that basically spiral around the cell, as you can see internally. And the spirochetes are the ones that are famous for having those endoflagella. Now, the endoflagella, they would produce a movement similar to the way a corkscrew would move through a cork. So that's kind of how the bacteria would move, because it has that axial filament. So that's just another image right here showing you the axial filament inside of the organism right here. Now, some bacteria have what's known as Fimbriae, and those Fimbriae, as you can see, they would surround the surface of the bacteria. Those Fimbriae, they're hair-like appendages, and they allow for attachment. They basically have a tendency to adhere they would allow the bacteria to adhere to other bacteria, or they would allow the bacteria to adhere to surfaces. And so having Fimbriae definitely makes the bacteria involved in forming biofilms. And that's when bacteria stick to surfaces, kind of like your teeth, rocks, so many different surfaces bacteria can attach to. And having those Fimbriae, again, can help the bacteria basically attach to all of these different surfaces. They can even allow the bacteria to even attach to the lining of your small intestine. So E. coli actually has those Fimbriae around its surface, and therefore E. coli can actually stick to the lining of your small intestine and cause diarrhea. When those Fimbriae are absent, usually no disease would occur, because you wouldn't have colonization, because having those Fimbriae will definitely help the bacteria colonize pretty quickly and grow pretty quickly on the surface. And definitely if they are colonizing your insides, that can cause problems. Some bacteria have what's known as pili, and those pili, they're a bit longer compared to the Fimbriae, and those pili, they are involved in motility, so like they can help the bacteria move. Another thing is that those pili can actually help the bacteria transfer its own DNA to another bacteria. So let's say here are two bacteria right there. Notice how they're both sticking out their pili, and what happens is that their pili could actually connect, and it kind of creates a bridge. Notice how they are creating a bridge between them. And what's really interesting and cool about that is that this bacteria, let's say, is carrying an antibiotic resistant gene that this bacteria is not carrying. What's going to happen is this bacteria can actually take its antibiotic resistant gene, and it can actually transfer it over to this bacteria right there. And if this bacteria is carrying, again, a gene that, let's say, makes it resistant to heat, for example, it can also pass it on to the other bacteria. And so what happens is, when bacteria do that and they start exchanging DNA amongst one another, if we are trying to, let's say, get rid of those bacteria, it's going to be a very hard task for us, because those bacteria are going to be changing on us very quickly, since they are exchanging their DNA and basically getting new characteristics and new properties that they didn't have before. So it makes it, again, a harder job for us to get rid of those pathogenic bacteria, because as I said, they could actually start exchanging antibiotic resistant genes. Now, if we talk about the structure of the cell wall of the bacteria, there are differences between the cell walls of different bacteria, and we can actually figure out those differences and figure out the type of cell wall that the bacteria is carrying by doing certain stains, like a Gram stain can tell us some differences between the cell walls of the bacteria. Having a cell wall actually contributes to the virulence of the bacteria, and we're going to talk about how. The cell wall of a bacteria is made up of something known as peptidoglycan. Peptidoglycan is basically consists of disaccharides, which are basically sugars, and polypeptides, which are proteins. Having a cell wall can actually prevent the bacteria from osmotic lysis, which basically means it can prevent the bacteria from intaking too much water. So it can prevent the bacteria from basically swelling up and bursting. So those are the main components of the cell wall. As I said, the cell wall, it's made up of disaccharides. So those rods that you're seeing right here, those are the disaccharides. Those are basically the sugar portion. And then those blue kind of greenish beads that you are seeing, those are representing the polypeptides. Those are the proteins. Those are the sugars that the peptidoglycan is made out of, and again, they're represented by those rods right here, and then those are the polypeptides. Now, when we look at a gram-positive cell wall, the gram-positive cell wall has many layers of that peptidoglycan versus the gram-negative. They only have a very thin layer of that peptidoglycan. So right here, if this is a gram-positive bacteria, it would have, first of all, a plasma membrane. On top of the plasma membrane, you would find the cell wall. So as you can see, the cell wall of a gram-positive, it's definitely made up of several layers of those disaccharides and the polypeptides. So you can see it's pretty thick. Gram-positives also has gram-positive bacteria also has those acids right here known as teichoic acids and lipoteichoic acids. Those teichoic acids, they basically provide they act as an antigen, meaning that they're pretty specific. So each bacteria could have slightly different teichoic acids. And so therefore they act as a self-antigen for the bacteria. And basically that antigen is going to note that this bacteria is kind of different from the other bacteria, depending on which exactly type of teichoic acid is it carrying. And so it could actually make it possible for someone to identify exactly which gram-positive bacteria based on the teichoic acid that it's carrying. Because again, as I said, it can work or it's going to act as a self-antigen for that bacteria. Biggest difference between a gram-positive and a gram-negative is definitely that cell wall structure. So first of all, there would be a plasma membrane, and then notice how the gram-negative only has one layer of that peptidoglycan. So that's why we say gram-negative. They have a very thin peptidoglycan. But what else do they have? On top of the peptidoglycan, they have what's known as an outer membrane. And this outer membrane consists of some polysaccharides, and it consists of lipids, and we call that lipid A. Now, having that outer membrane within the gram-negative can actually help the gram-negative bacteria evade phagocytosis. Having that outer membrane can actually help the gram-negative bacteria create kind of like a barrier. So that outer membrane kind of acts as a barrier to antibiotics, for example, penicillin. It acts as a barrier against digestive enzymes. And so you can see that when gram-negative bacteria have that outer membrane, you can see how much that outer membrane is basically going to be helping out that gram-negative bacteria evade the immune system of the host, evade basically getting broken down by enzymes that the host again might throw at that bacteria right there. Another thing that's going to, help out the bacteria is that lipid. Lipid A is basically that lipid portion of that lipopolysaccharide, and that lipid, as you can see, it is embedded in the outer membrane of the gram-negative bacteria. And so what happens is when the bacteria, let's say a gram-negative bacteria dies, when that bacteria dies, they actually release their lipid A, which is going to act as an endotoxin. And so you can see that even if the host succeeds in killing a gram-negative bacteria, that gram-negative bacteria, if it's killed, it's actually going to release that lipid A, and that lipid A is going to, it's basically a toxin to the host. And so that lipid A is definitely responsible for a lot of the symptoms associated with getting infected by a gram-negative. Because again, as the host immune cells are trying to protect the host, and they're going to do their best trying to kill that gram-negative bacteria. If some of those gram-negative bacteria, they start dying, they're going to release that toxin, which is the lipid A, and some of the symptoms that you would, for example, have is fever, shock, dilation of your blood vessels, blood clotting. So you can see it's a huge list of negative side effects that the individual would face if that lipid A, again, is released. Another thing attached to that lipid A, there's this O polysaccharide, and that O polysaccharide basically acts as a self-antigen. So it kind of acts as an antigen that's going to help us identify this bacteria. So it would help us distinguish which gram-negative bacteria we are talking about. And so that O polysaccharide is kind of acting the same as the teichoic acid in gram-positive bacteria. So the teichoic acid in gram-positive and the O polysaccharide in the gram-negative, they kind of have the same function, which is, again, they would help us identify which type of bacteria we are talking about. Now, bacteria, they do have DNA, but other than having that circular thread of DNA, they also usually have what's known as plasmids. And those plasmids, they are basically extra chromosomal DNA, meaning that they are not part of the original DNA. And those plasmids, they usually carry genes for things like antibiotic resistance, tolerance to toxins. Those plasmids can even be transferred from one bacteria to another, again, via those pili that we've talked about previously. And so therefore, those plasmids are pretty important, and they are definitely beneficial if a bacteria carries those plasmids. Bacteria, they also do carry ribosomes, and those ribosomes, they are basically how the bacteria is going to be able to make its own protein. The bacteria relies on its ribosomes. Ribosomes are made out of proteins and some ribosomal RNA, and a ribosome would look something like this. It has two subunits, and the subunits for the ribosomes of a bacteria, we call it 50S and 30S. And so collectively, we say that ribosomes have a bacteria known as the 70S bacteria. By the way, that 70 is not the addition of the numbers 30 plus 50. That would give you 80. So again, those subunits, you don't add them. It's just, again, this one's called 50, this one's called 30, but the whole thing is called 70. Now, when essential nutrients are basically getting depleted from a bacteria, the bacteria is going to go ahead and start producing what's known as an endospore. Endospores are usually produced by gram-positive bacteria, such as the ones under the genera or genus Bacillus and Clostridium. So those are usually the ones that produce those endospores. Endospores, they are basically more durable, kind of daughter cells, if you can think of it this way. And those endospores, they have very thick walls that make them basically able to tolerate high heat. They're able to tolerate like harsh environmental conditions. Those endospores, they're made internally within the bacteria cell membrane, and they can actually get released into the environment. And once they are released into the environment, they can actually survive on their own. And as I said, they can survive extreme heat, they can survive lack of water, they can actually tolerate a bunch of toxins and radiation. So as you can see, getting rid of the endospores is not an easy task. They've actually found endospores that were 40 million years old, and when they took that endospore back to the lab and they've given it the nutrition that it needs, it was able actually to germinate back. So let's talk about the process of forming those endospores, and that process is known as sporulation. But first, let me kind of show you an image. So this is a bacteria forming the endospore inside of it. So one more time, the endospore is going to be developed inside of the bacteria. So as I said, when a bacteria is missing, or the major important nutrients that bacteria needs is starting to become scarce, the bacteria is going to feel the urge that it's time to go ahead and create those endospores. Or if the bacteria feels that it's threatened, like it's not getting enough nutrition, it's going to go ahead and start producing those endospores. And so what happens is the bacteria is going to start replicating its own DNA. So it's going to start making extra copies of its own DNA. So let's say here's the original DNA. It's basically going to replicate it. So here's the replicated DNA. And then as you can see, the plasma membrane starts to actually enclose around that that newly replicated DNA. So you can kind of see it's encapsulating it right there, and you can see that it's going to continue encapsulating it to the point that it's kind of like, you know, separating it from the rest of the bacteria. So within this capsule, you have the DNA, you have some of the cytoplasm, and here is the plasma membrane. Now, what happens is that you start getting like two membranes. So the plasma membrane kind of, you start getting like those two membrane layers of plasma membrane, and then what starts happening is the peptidoglycan is actually going to start forming between those membranes, and then the bacteria itself is actually going to burst open to allow the spore to actually go ahead and get released. And so the endospore is going to get released. Now, once that endospore gets you know, nutrition, and it feels like, okay, now the environmental condition that it's within is okay, and it's going to be able to support its growth, the endospore is basically going to go ahead and germinate one more time. But those endospores, they can actually stay dormant for thousands of years. And then, as I said, they found an endospore that was 40 million years old, and after giving it the nutrition that it needs, it was actually able to germinate one more time. Endospores can survive very harsh conditions. So it would be a problem if there are endospores in food or in like canned food, for example, because it's extremely hard to get rid of them. You would think, okay, why not just kind of like boil it? Well, it turns out that some of those endospores can actually survive in the boiling water for 19 hours. So again, you can kind of see how hard it is to get rid of those endospores.